So hopefully advising day went well for everybody. We have another small class today, which makes me a little sad, but also I love teaching to smaller class sizes, so it's pros and cons here. Any questions about materials, what class has been going, anything in general? All right, my goal is to get the study guide for the next exam up by Thursday or Friday, by the end of this week. That's my goal. But I don't know if you've noticed at the end of the slides, I've been including like the mini study guides. So the main study guide is basically just all those main ones put together. But I want to go through and make sure that the study guide is all encompassing and do some checks on it. So that's my goal. Let's see here. I did change a date in the schedule. All right. So Wednesday before Thanksgiving, I had on there that we had class. No classes are happening that day. All right. That was an error on my part. All right. You actually don't have class that day. I thought you did, but I put it down as no class day. All right. So that is a minor change. We will catch up from any materials that we were supposed to go over eventually. All right. Let's move into some transcription and translation. So on, let's see, Monday now, we talked about gene expression and how we can express different proteins in different cells, and that's how we get a lot of our diversity in our cells. But I wanted to go on and talk about how this actually gets expressed. So we talked about that central dogma of biology, and that's where we left it on Monday. So that central dogma is taking DNA, turning it into RNA, and then translating that into a protein that has a function, right? And so transcription and translation are the process from going to DNA to RNA and RNA to a protein is really how we're turning these genes on and off, really how gene expression is happening, right? And so we're going to look at transcription first today. And I think we'll be able to begin talking about translation and then finish this up on Friday. So we're going to dive right on into transcription, right? But in order to talk about transcription, there is some terminology you need to know associated with our genes and with our DNA. So first of all, if we look at a segment of DNA that will be transcribed into RNA, there's these different regions. All right, so one of those regions is the regulatory region. It shows up as this green and like off-color red here, right? This region really controls how fast transcription might happen. Right? It also acts as an area where we have this promoter, right? And the promoter is the site where DNA transcription actually begins, right? So this regulatory region, that's telling us how we might affect transcription rate, right? This promoter region is saying this is where we are actually going to start transcription. And then at the other end of the section of DNA that will be transcribed, we have this termination region or the terminator site, right? And this is the site that tells transcription to stop. Right? This is where transcription of our DNA will end. Right? So if we look at this segment of DNA that we will transcribe, we have a region that tells us how fast. We have a region that says this is where we're going to start. Right? And we have a region that says this is where we're going to end. Now in between that start and stopping is where we get our gene. Right? So the actual information that will be encoded and translated into a protein. All right. Genes tend to be about 10,000 to 50,000 base pairs long, right? Though they can be way longer than that. Right? And genes can include two regions when we first transcribe that DNA, right? The first is going to be an exon, right? So an exon is a sequence that is going to be kept in the final RNA product that will be made in transcription. You can think of it as the expressed regions, so exons. But we also have these introns, right? These introns are regions that will be transcribed, but it will eventually be removed in that final RNA project, right? And we can think of that as these intervening regions here, right? And on our image here, we have these exons in this light blue and these introns in these yellows, right? When we get to the end of transcription, these introns will be removed and they will not be transcribed into a protein. All right, so we have some of that terminology down. Let's move right on into transcription here. All right, so transcription is the process of making an RNA copy. Right? And specifically, you might see it listed as mRNA. That stands for a messenger RNA. Right? And we'll dive into that a little bit deeper in a few slides here. Right? But we are making an RNA copy of the gene using DNA as our template. Right? So if we look at transcription, there are 3 stages. Right? We have initiation, we have elongation, and we have termination. Right? This is a beautiful image up here, right? The text doesn't come through very well, but I did leave this text whole if you go to some of the slides at the very end of this PowerPoint lecture here. So we'll walk through each of these stages, and we'll start with initiation. Now for transcription, I'm going to walk us through mostly how bacteria do it, right? But I will also tell you how eukaryotes differ slightly, but I want you to know more of the specifics of bacteria, right? So if we look at initiation, right, we have our segment of DNA that is going to be transcribed, right? The first thing that will happen is we get this transcription factor called sigma factor, right? It's a protein, and it's this protein that's going to find our promoter region, right? It's that region that tells us to start DNA transcription, right? So we have our sigma factor here. It's finding that promoter region, which is blue in our diagram here. Right? Once it finds that promoter region, an enzyme is going to come in called RNA polymerase. Right? That's going to bind to the sigma factor, and it's going to start prying the strands of DNA apart. Right? So similar to how DNA polymerase might work, right, with the helicase aiding it, right? But this time, RNA polymerase can unwind it in itself. Right? When it unwinds this DNA, so our RNA polymerase here is this nice big purple protein, right? It's going to form what's called an open complex. Right? So an open complex is this region that has been unwound, right? This region of DNA that has been unwound here. Right? And if we look at this region of DNA, we have the coding strand. We also have a template strand. Right? We only need one strand of DNA to make an mRNA molecule. Right? So that mRNA molecule is going to be made off of that template strand. Now this open complex is only about 10 to 15 base pairs in length, so it's relatively short. Right? If we look at how eukaryotes do this step, it's basically the same, but with a lot more proteins involved, right? So instead of having a single transcription factor like bacteria does with that sigma factor, right, they have multiple factor proteins. They also have multiple RNA polymerase types, right? And those multiple RNA polymerase types will locate and bind to the promoter region and transcribe different things, right? So a little bit more complex in eukaryotes. If we look at the next step, right, the next step is elongation here. So this is where we are actively making that RNA molecule. We are transcribing the data in our DNA into an RNA molecule. Right? This is always going to happen in the 5 to 3 direction, just like DNA polymerase works. Right? And as RNA goes through and transcribes this genetic material here, remember this is going to be transcribed into RNA, which means any time this RNA polymerase comes across an A or an adenosine here, instead of a thymine, it has to add in the uracil, right? Because RNA does not contain thymine. Okay? So during elongation here, we're going to add and create this RNA strand. Our sigma factor is going to be released, right? We no longer need our sigma factor. It is going to go away, in eukaryotes, those multiple factors will be released, right? And as elongation happens, our RNA polymerase is just going to slide along our DNA molecule, maintaining that open complex as it transcribes that information, right? So as this open complex transcribes this information, it's just going to keep moving along, right? And it's actually going to, that DNA is actually going to rewind itself after it's been transcribed. So it's not like the replication bubble where we split apart two strands and we make two new strands, right? This time we're just kind of opening it, doing what it needs to do, and then it's closing back up, right? So again, nucleotides, they're synthesized in that 5' direction. This is basically the same image, a different way of showing it here, right? We are making our RNA molecule, and once it's being made, the DNA will rewind into that double helix. All right, the last stage is the termination stage, and it is the easiest stage to understand because, simply put, right, everything's going to come off of our DNA strand, and we have a complete RNA molecule, right? So RNA polymerase has reached that terminator sequence, right, the sequence telling it to stop transcribing, and the polymerase is going to fall off, right, as well as that completed mRNA transcript. All right, let's do some review here and step back. I want you to list what happens in each stage, right? And what is the name of the protein used in transcription, the protein that actively synthesizes the new RNA strand? I forgot to put timers on these slides, so I will just read the room and see when we are ready. I know, I'm doing more information. Yeah. So they think the Tron might slicing things up. Was the talk about slicing this before? Neutron might it's a while ago we thought neutrons have but we know they have a purpose, but we're still discussing. All right, what happens in the initiation stage? Yeah, so initiation, right? The sigma factor is finding that promoter region, and that, like Isabella said, lets that RNA polymerase know where to start, right? Also, that RNA polymerase, right, once it binds that sigma factor, knows where to start, is going to form that open complex, right? That region of DNA that has been unwound, a little unwound bubble, if you will. All right, so that's initiation. What happens in elongation? What's being actively made? What's growing? What's elongating? RNA, yes, we are making RNA, actively making that RNA strand, that mRNA, right? The sigma factor is released, right? Nucleotides are added in that 5 to 3 direction. Right? Directionality is always important to talk about when we're talking about DNA or RNA. And that DNA is going to rewind after it's been read. And that last step, termination, what's going on? I heard it. It's terminated, right? Yeah. It ends. It stops. And the proteins that were used and the RNA molecule that was made is going to disassociate from the original DNA strand, right? And the name of that protein used in transcription, it's RNA polymerase, which we have already mentioned here. There's that other protein, the sigma factor, as well. All right. So that's not the end of transcription. There is still more that has to be done for eukaryotes. Right? For bacteria, this is great. They're done. They can move on to the next step. For eukaryotes, we have to process this, what's called a pre-mRNA strand, before it becomes a mature mRNA strand. Right? So essentially, we have to get rid of those introns that we had talked about previously. Right? So we have to get rid of those introns that they were transcribed, right? But we're not going to translate them into protein. So we have to get rid of them while keeping those exons. Right? The exons are those regions that are actively transcribed into that protein. And so if we look at the final product here, we'll notice that those introns, which show up as yellow here, are completely gone. Right? This process is called splicing. Right? But again, this is not the end of it. We still have to add in some more modifications. So besides removing those introns, we are also going to add a cap and a tail onto our RNA molecule. Right? And so if we add in that cap, right, we are adding basically a modified nucleotide, a modified guanosine, to the 5' end of our mRNA. So if we look at our modified guanosine here, we can see it looks kind of similar, right? It has a sugar, it has our nitrogenous base here that's been slightly modified, and it has a couple of extra phosphate groups. Right? We're adding it to the 5' end, and the purpose being is that we need to move this RNA molecule from the nucleus, where it has been transcribed, into the cytosol, where it's going to be translated. Right? And this is really why it's called messenger RNA, because it can move between the nucleus into the cytosol here. We're also going to add in this poly-A tail, right? It's called the poly-A tail, poly meaning many, and the A stands for adenosine. Right? So we are adding a bunch of our adenosine nucleotides to the 3' end of our RNA molecule, right? And this does a couple things. Right? It's going to increase the stability of our mRNA molecule and increase its lifespan in the cytosol. Right? That way, it has time to be translated before it can dissociate and be recycled into newer molecules. Right? It's important to note that even though we're adding in these nucleotides, right, they were not nucleotides that were encoded for in the original DNA sequence. Right? They are added in. You can kind of say it was kind of an afterthought, even though it has a very clear purpose to why we are adding this to the 3' end. All right, another class question here. I'm putting in lots of stop points because I know this is a lot of information. So I want you to tell me what 3 things need to be modified in this sequence of in DNA, I should say, when it's being made into mRNA. And does RNA processing occur in prokaryotes? All right, how is this gene going to have to be modified when it's made to a mature RNA molecule? What's one of the first things that's going to happen? Yeah. It's going to get spliced, yes. We got to remove those introns. What else has to happen? Yes, we're splicing, we are capping, and we are adding in that poly A tail. Yes. All right, so 3 things have to happen for this to become a mature mRNA molecule. Does RNA processing happen in prokaryotes? All right, hands up if it's yes. Yeah, go for it. They will be processing. They're already ready to go. Right? Bacteria cells, they don't have to add a tail and a cap. Bacteria actually don't even have introns, so we don't have to go through any splices. So yes, this only happens in eukaryotes, and that's an important differentiation here. Any other questions? Hannah asked a good question earlier about introns and why we even have them if we're going to remove them. And short answer is they have a function and we are still figuring it out. All right. We used to think they didn't have a function, but now we know that they do, and it could be in making alternate proteins. It could be in helping splicing up the gene in different ways. We are actively researching it. All right. So, back to the central dogma. We've gone over transcription. We now have to go over translation. All right, so we got to take this RNA molecule and make it into a protein that has a function that actually does something in our body. All right, so translation. All right, this is the process of synthesizing a polypeptide. All right. So if you think back to, the first month of class, we talked about polypeptides. All right, so that is a sequence of amino acids, right? It's not a functional protein, right? It's just that sequence of amino acids. Once that sequence is made, then it will fold into those different three-dimensional shapes and become functional. Right? So translation is just making that sequence of amino acids, that linear sequence. This occurs in the cytosol, right? So we are no longer in the nucleus. We have moved into the cytosol, and it's specifically going to happen at ribosome. Right? So the ribosome is the key player here. Right, so in the cytoplasm, at a ribosome. All right, in order to understand translation, before we even get into the mechanisms of how it works, we have to understand the genetic code, right? So the genetic code is the sequence of bases in a messenger RNA molecule. Right? If we looked at this molecule and we wrote out all the letters in order, that would be the genetic code. Right? Now we read this genetic code in groups of 3 nucleotides. And those groups of 3 would be called codons. Right? These codons, they are going to specify a particular amino acid. Right? So codon 1, 2, 3, 4, 5, 6, 7, they will all be different amino acids that will be added on in the order. I shouldn't say different, they could be different. Right? We'll see why what I just said might be slightly incorrect in the next few slides here. Right? These codons can also specify start and stop codons. Right? So where to start making the amino acid chain and where to stop making the amino acid chain. All right, so if we are trying to translate this section of mRNA, right, it's always going to be read from the 5' to the 3' end. Right? And we can use this nice codon chart here to figure out what the amino acid would be. So on this chart, we have a series of 3 letters, right? And in those 3 letters, it's going to tell me what amino acid it is coding for in a shorthand form here. Right, so let's look at our mRNA here. First, we want to break it into codons. So split it into groups of 3. All right. After we split it into groups of 3, you're now going to correlate our groups of 3 to our chart to figure out what amino acids will be added. So AUG, right? If I look for AUG here, right, I find AUG here, right, and this is methionine, right, but it's also a start codon. So AUG is a good codon that says start synthesizing the polypeptide here with this amino acid, right? So it's a start codon, but it also codes for methionine. But I like to look for my next one here, which is UCU. Right? I can go to U. I know the second letter is C. UCU is serine here. Right? And I would keep going down the list until I have translated all of my codons into amino acids. Now, you might notice it doesn't quite line up. There's this last one here, right? That does not code for amino acid. And if we look at our chart, this UAA, let's see, UAA, you see that that's a stop codon. Right? This is telling translation to stop making that amino acid. Right? Unlike the start codon that has amino acids associated with it, the stop codon does not. Right? We're not going to put an amino acid there. We're simply going to stop. All right, so another opportunity for you to put it into practice here. I am giving you a strand of mRNA. I want you to first separate it into codons and then translate it into amino acid sequences, or a sequence. This is always one of my favorite things to do in bio. It was easy to me and it made sense. We can make a sentence out of this. I bet people have. gotten to a point where you would separate them out into three. And eventually, you're going to put them into a sequence of amino acids. Now, what I did not tell you is what did you all put for your first amino acid? Oh, yes. And that is in there kind of explicitly, right? I would not discredit you if you had put down pro first. The thing is proline, if I'm remembering the full version of an amino acid, but it's before that start codon, right? So because it's before the start codon, it actually won't be translated. So, won't be translated, which is why we don't have it down here. We will start with that methionine and go all the way down until we get to our stop codon. Right. Any questions on this? We will keep charging forward then. So if we're looking at our genetic code, there are some important properties of this code. Right? First of all, it's redundant. Right? So all amino acids are coded by more than one codon. Right? Sometimes 4, sometimes 2. Right? But it's going to have more than one codon that can code for an amino acid. Okay. It's unambiguous, right? So a single codon is always going to code for a single amino acid. It's never going to code for more than one amino acid. It's non-overlapping, which means we read it in distinct segments of the. So if we had a sequence, let's go AUG of our starts, then I'll just throw some letters up here. Right? You would read it in sets of 3. The second letter of a set of a codon is not going to code for another codon. Right? It's not going to overlap this. So that's what I mean by non-overlapping. This is not going to happen. And it's conservative, which means that the first 2 letters are almost always going to be the same for a amino acid. So the first 2 bases are almost always the same. There's some variations, right? If you look at I'm not going to try to remember that name there, but TYR here, right? In this case, that second letter is not. Right? But typically, the first 2 letters are always going to be the same, right? And then it's that last letter that will vary for a particular amino acid. And this is important, right? Because if mutations arise, right, it's less likely to change what that amino acid is when that mutation happens in a third spot. Right? So it's conservative in that manner. All right, so we know what a codon is. We know what our mRNA is, but there are more RNA molecules involved in translation. Right? So mRNA contains our codons, our sets of 3 RNA nucleotides, right? Where we have T substituted for U. But we also have this RNA called tRNA. Right? This tRNA is going to contain an anticodon. This anticodon is complementary to the codons in mRNA, right? And so it allows for the binding of our tRNA molecules to our mRNA codons, right? And this is important because our tRNA molecules carry those amino acids that are coded for by our codons, right? So we have our mRNA, we have our tRNA. And if we look at, get a big picture here, we can see we have gone from our DNA strand, all right, our double helix DNA here, though it's not in helix format in this diagram. It has been transcribed into mRNA, right, single-stranded here. Our mRNA has codons, or these sets of 3s, right, that are complementary to the anticodon in these tRNA molecules. Right? And these tRNA molecules carry those amino acids, which will make up the resulting polypeptide. All right, so this tRNA molecule has some features to it. First of all, they'll be encoded by different genes depending on the different types of tRNA molecules. The tRNA molecules will also be named according to the amino acid that it carries. So for example, this tRNA molecule, we have the superscript SER because it carries serine. Right? If it's not carrying anything, it'll just be tRNA. And there's some common structural features. So first of all, it has this cloverleaf feature. Right? So if we look at this two-dimensional structure, it has this cloverleaf to it. It has what's called stem regions, right, where it actually becomes base paired together through hydrogen bonds. Right? We don't often see that in RNA, but in our tRNA molecules, there is some base pairing occurring in those stems. We also have these loops, or these, loops, right, where base pairing is not occurring. Right? If we look at this in a three-dimensional structure, it starts to get a little less clover-like shapes, all right, but there are still regions of base pairing occurring. We are going to have an anticodon, right? That's always going to be on one of these loops, right? That middle loop here. So our anticodon, that's going to directly interact with our mRNA. And we have an acceptor stem. So the acceptor stem is a region at the 3' end. It is a single-stranded region, so it is not base paired at this area. And this is where the amino acid will attach to the tRNA molecule. All right, so how does it attach to the tRNA molecule? All right, we're not going to go into any specifics, but I want you to give you the broad picture. All right, so we're using an enzyme called aminoacetyl tRNA synthetase. Right? The word synthetase doesn't feel right in my mouth when I say it. Right? But this synthetase is catalyzing the amino acids attaching to our tRNA. So essentially, we have our enzyme. Our amino acid is going to find the active site specific for that amino acid. ATP is also going to bind, right, because it takes energy for this to happen. ATP is essentially going to allow this amino acid to stay in place while this tRNA molecule comes in and binds to this enzyme. Right? And this enzyme will then catalyze the reaction of putting that amino acid onto the tRNA molecule. When it has an amino acid on it, we would say it's a charged tRNA, right? Charged tRNA, meaning it's charged up, it's ready to go, we can put it to work. All right, yet another checkpoint for us here. So, tRNAs, super important. I'm sure you will see them again in your careers, all right, in biology here. So describe that general structure using this image, right? And then contemplate, right, what is the functional importance of that aminoacetyl tRNA synthetase, and what would happen if that enzyme became defective? What's the general structure of this tRNA molecule? What do we say it looks like? Yeah, we say it looks like a clover. It has this clover leaf structure with roots and stems. Stems are where we get some of this base pairing occurring. What's shown in region A up here? It's called the acceptor stem. That's where the attachment site is located for the amino acid, right? Which means B, right, would be what? Anticodon, all right? And so if we think about our tRNA molecule here, what would happen if the enzyme that puts the amino acid on this tRNA molecule becomes defective? What could be some important thing that might be not good? I do wish we were over here. You want to share, I think?
That enzyme helps the amino acid attach to the and if the enzyme attaches, then the long part enzyme tends to be
Yeah. If we can put an amino acid on these tRNA molecules that are used in translation, right, we're not going to synthesize proteins, right, because the amino acids can't attach to the tRNA molecule that brings it to the mRNA. Any questions? All right. I didn't think we would get this far. I'm actually kind of impressed. Let's talk about ribosomes and maybe do a couple more slides. And we're a little early today. So, translation happening at ribosomes. Right? Ribosomes in prokaryotes, they only have one type of ribosome. In eukaryotes, there's distinct ribosomes in different organelles. So if you think about mitochondria and chloroplasts, they both have ribosomes in them. Right? But when we're talking about translation, we're going to focus on the ribosomes that are in the cytosol or in that rough ER. All right. Ribosomes are composed of two subunits. Right? So one subunit is the small subunit. The other subunit is the large subunit. And you can kind of see it in this 3D rendering here. Our small subunit is this skinny piece here, and the large subunit kind of has more of a dome shape to it on the other side. Right? So those two subunits will eventually come together when we make our protein. Right? We also have another type of RNA molecule in here. We have rRNA, or ribosomal RNA, right? Ribosomal RNA plays an important role in giving the ribosome its shape and structure. Right? So that's our third type of RNA that we have listed today, right? We have messenger RNA, tRNA, and rRNA. Right? Now, one cool little note here is that we notice there's some differences between bacterial ribosomes and eukaryotic ribosomes, right? And antibiotics can actually exploit this difference, right? So that they can target specifically bacterial ribosomes rather than the eukaryotic ribosomes, right? So if we have a bacterial infection, antibiotics can exploit the difference between these structures here. So, kind of an interesting fun fact, right? We look a little bit more in depth in the structure of the ribosome, and I think we'll stop after this slide here, that there's 3 sites we want to be aware of, right? And these 3 sites are important for translation. The first is the P site, or the peptidal site, right? This is where the growing peptide chain is going to be, right? That's the middle site in our ribosome here, right? Important to know that the site is on our large subunit here, or a top half of the ribosome. We have this A site, this aminoacetyl site. This is where the tRNA molecule will initially come into the ribosome. And then we have this E site, right, or the exit site. And that's where once that tRNA molecule gives its amino acid to the growing polypeptide chain, it will leave on the E site. All right. Any questions? All right, this is a really great stopping point. So I'm going to stop us here, right, and we'll pick up on this on Friday. Keep in mind, Friday we do have another content check, which I will double check, but I think we've gone through all the material that will be on that content check. But I will go through and I'll send out an email if we need to edit it at all. All right. Scientific literacy assignment is not due until November 17th. I should get that posted later today if you want to work ahead.